Section 49 of Gray's Anatomy, Part 4. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Read by Lauriann Walden. Anatomy of the Human Body, Part 4, by Henry Gray. The Organ of Sight. Organon visus, the eye. The bulb of the eye, bulbus oculi, eyeball, or organ of sight, is contained in the cavity of the orbit, where it is protected from injury and moved by the ocular muscles. Associated with it are certain accessory structures. These are the muscles, fascia, eyebrows, eyelids, conjunctiva, and lacrimal apparatus. The bulb of the eye is embedded in the fat of the orbit, but is separated from it by a thin membranous sac, the fascia bulbi. It is composed of segments of two spheres of different sizes. The anterior segment is one of a small sphere. It is transparent and forms about 1/6 of the bulb. It is more prominent than the posterior segment, which is one of a larger sphere, and is opaque and forms about 5/6 of the bulb. The term anterior pole is applied to the central point of the anterior curvature of the bulb, and that of posterior pole to the central point of its posterior curvature. A line joining the two poles forms the optic axis. The axes of the two bulbs are nearly parallel, and therefore do not correspond to the axes of the orbits, which are directed forward and lateralward. The optic nerves follow the direction of the axes of the orbits, and are therefore not parallel. Each enters its eyeball 3 mm to the nasal side and a little below the level of the posterior pole. The bulb measures rather more in its transverse and antero-posterior diameters than in its vertical diameter, the former amounting to about 24 mm, the latter to about 23.5 mm. In the female, all three diameters are rather less than in the male. Its antero-posterior diameter at birth is about 17.5 mm, and at puberty from 20 to 21 mm. Development. The eyes begin to develop as a pair of diverticula from the lateral aspects of the forebrain. These diverticula make their appearance before the closure of the anterior end of the neural tube. After the closure of the tube they are known as the optic vesicles. They project toward the sides of the head, and the peripheral part of each expands to form a hollow bulb, while the proximal part remains narrow and constitutes the optic stalk. The ectoderm overlying the bulb becomes thickened, invaginated, and finally severed from the ectodermal covering of the head as a vesicle of cells, the lens vesicle, which constitutes the rudiment of the crystalline lens. The outer wall of the bulb becomes thickened and invaginated, and the bulb is thus converted into a cup, the optic cup, consisting of two strata of cells. These two strata are continuous with each other at the cup margin, which ultimately overlaps the front of the lens and reaches as far forward as the future aperture of the pupil. The invagination is not limited to the outer wall of the bulb, but involves also its postero-inferior surface and extends in the form of a groove for some distance along the optic stalk, so that, for a time, a gap or fissure, the choroidal fissure, exists in the lower part of the cup. Through the groove and fissure the mesoderm extends into the optic stalk and cup, and in this mesoderm a blood vessel is developed. During the 7th week the groove and fissure are closed, and the vessel forms the central artery of the retina. Sometimes the choroidal fissure persists, and when this occurs the choroid and iris in the region of the fissure remain undeveloped, giving rise to the condition known as coloboma of the choroid or iris. The retina is developed from the optic cup. The outer stratum of the cup persists as a single layer of cells which assume a columnar shape, acquire pigment, and form the pigmented layer of the retina. The pigment first appears in the cells near the edge of the cup. The cells of the inner stratum proliferate and form a layer of considerable thickness from which the nervous elements and the sustentacular fibers of the retina, together with a portion of the vitreous body, are developed. In that portion of the cup which overlaps the lens, the inner stratum is not differentiated into nervous elements, but forms a layer of columnar cells which is applied to the pigmented layer, and these two strata form the pars ciliaris and pars iridica retinae. The cells of the inner or retinal layer of the optic cup become differentiated into spongioblasts and germinal cells, and the latter, by their subdivisions, give rise to neuroblasts. From the spongioblasts, the sustentacular fibers of Müller, the outer and inner limiting membranes, together with the groundwork of the molecular layers of the retina, are formed. The neuroblasts become arranged to form the ganglionic and nuclear layers. The layer of rods and cones is first developed in the central part of the optic cup, and from there gradually extends toward the cup margin. All the layers of the retina are completed by the 8th month of fetal life. The optic stalk is converted into the optic nerve by the obliteration of its cavity and the growth of nerve fibers into it. Most of these fibers are centripetal and grow backward into the optic stalk from the nerve cells of the retina, but a few extend in the opposite direction and are derived from nerve cells in the brain. The fibers of the optic nerve receive their medullary sheaths about the 10th week after birth. The optic chiasma is formed by the meeting and partial decussation of the fibers of the two optic nerves. Behind the chiasma the fibers grow backward as the optic tracts to the thalami and midbrain. The crystalline lens is developed from the lens vesicle, which recedes within the margin of the cup and becomes separated from the overlying ectoderm by mesoderm. The cells forming the posterior wall of the vesicle lengthen and are converted into the lens fibers, which grow forward and fill up the cavity of the vesicle. The cells forming the anterior wall retain their cellular character and form the epithelium on the anterior surface of the adult lens. By the 2nd month the lens is invested by a vascular mesodermal capsule, the capsula vasculosa lentis. The blood vessels supplying the posterior part of this capsule are derived from the hyaloid artery; those for the anterior part from the anterior ciliary arteries. The portion of the capsule which covers the front of the lens is named the pupillary membrane. By the 6th month all the vessels of the capsule are atrophied except the hyaloid artery, which disappears during the 9th month. The position of this artery is indicated in the adult by the hyaloid canal, which reaches from the optic disc to the posterior surface of the lens. With the loss of its blood vessels, the capsula vasculosa lentis disappears, but sometimes the pupillary membrane persists at birth, giving rise to the condition termed congenital atresia of the pupil. The vitreous body is developed between the lens and the optic cup. The lens rudiment and the optic vesicles are at first in contact with each other, but after the closure of the lens vesicle and the formation of the optic cup, the former withdraws itself from the retinal layer of the cup. The two, however, remain connected by a network of delicate protoplasmic processes. This network, derived partly from the cells of the lens and partly from those of the retinal layer of the cup, constitutes the primitive vitreous body. At first these protoplasmic processes spring from the whole of the retinal layer of the cup, but later are limited to the ciliary region, where by a process of condensation they appear to form the zona ciliaris. The mesoderm, which enters the cup through the choroidal fissure and around the equator of the lens, becomes intimately united with this reticular tissue, and contributes to form the vitreous body, which is therefore derived partly from the ectoderm and partly from the mesoderm. The anterior chamber of the eye appears as a cleft in the mesoderm separating the lens from the overlying ectoderm. The layer of mesoderm in front of the cleft forms the substantia propria of the cornea, that behind the cleft the stroma of the iris and the pupillary membrane. The fibers of the ciliary muscle are derived from the mesoderm, but those of the sphincter and dilatator pupillae are of ectodermal origin, being developed from the cells of the pupillary part of the optic cup. The sclera and choroid are derived from the mesoderm surrounding the optic cup. The eyelids are formed as small cutaneous folds, which about the middle of the 3rd month come together and unite in front of the cornea. They remain united until about the end of the 6th month. The lacrimal sac and nasolacrimal duct result from a thickening of the ectoderm in the groove, naso-optic furrow, between the lateral nasal and maxillary processes. This thickening forms a solid cord of cells which sinks into the mesoderm. During the 3rd month the central cells of the cord break down, and a lumen, the nasolacrimal duct, is established.